chime in on anything before we officially dive into the material? All right, so today we are going to be talking about learning, memory, and emotions and how that really relates to the human nervous system. So I'm going to have you watch this clip. I'll show it on my screen, but it's also on Blackboard, and that'll kind of kick us off. Then we'll move to the PowerPoint.
So how do we learn? And why do some of us learn things more easily than others? So as I was mentioned, I'm Dr. Laura Boyd. I'm a brain researcher here at the University of British Columbia. These are the questions that fascinate me. So brain research is one of the great frontiers in the understanding of human physiology, and also in the consideration of what makes us who we are. It's an amazing time to be a brain researcher, and I would argue to you that I have the most interesting job in the world. What we know about the brain is changing at a breathtaking pace. And much of what we thought we knew and understood about the brain turns out to be not true or incomplete. Now, some of these misconceptions are more obvious than others. For example, we used to think that after childhood, the brain really could not change. And it turns out that nothing could be farther from the truth. Another misconception about the brain is that you only use parts of it at any given time and silent when you do nothing. Well, this is also untrue. It turns out that even when you're at rest and thinking of nothing, your brain is highly active. So it's been advances in technology such as MRI has allowed us to make these and many other important discoveries. And perhaps the most exciting, the most interesting and transformative of these discoveries is that every time you learn a new fact or skill, you change your brain. It's something we call neuroplasticity. So as little as 25 years ago, we thought that after about puberty, the only changes that took place in the brain were negative. The loss of brain cells with aging, the result of damage like a stroke. And then studies began to show remarkable amounts of reorganization in the adult brain. And the ensuing research has shown us that all of our behaviors change our brain, that these changes are not limited by age. It's good news, right? And in fact, they're taking place all the time. And very importantly, brain reorganization helps to support recovery after you damage your brain. The key to each of these changes is neuroplasticity. So what does it look like? So your brain can change in three very basic ways to support learning. And the first is chemical. So your brain actually functions by transferring chemical signals between brain cells, what we call neurons, and these trigger a series of actions and reactions. So to support learning, your brain can increase the amount or the concentrations of these chemical signaling that's taking place between neurons. Now, because this kind of change can happen very rapidly, this supports short-term memory or the short-term improvement in the performance of a motor skill. The second way that the brain can change to support learning is by altering its structure. So during learning, the brain can change the connections between neurons. Now here, the physical structure of the brain is actually changing, so this takes a bit more time. These types of changes are related to long-term memory, the long-term improvement in a motor skill. Now, these processes, they interact, and let me give you an example of how. So we've all tried to learn a new motor skill, maybe playing the piano, maybe learning to juggle. You've had the experience of getting better and better within a single session of practice and thinking, I've got it. And then maybe you've returned the next day and all those improvements from the day before, they're lost. What happened? Well, in the short term, your brain was able to increase the chemical signaling between your neurons. But for some reason, those changes did not induce the structural change that are necessary to support long-term memory. Remember that long-term memories take time, and what you see in the short term does not reflect learning. It's these physical changes that are now going to support long-term memories and chemical changes that support short-term memories. Structural changes also can lead to integrated networks of brain regions that function together to support learning. And they can also lead to certain brain regions that are important for very specific behaviors to change their structure or to enlarge. And so here's some examples of that. So people who read Braille, they have larger hand sensory areas in their brain than those of us who don't. Your dominant hand motor region, which is on the left side of your brain if you're right-handed, is larger than the other side. And research shows that London taxi cab drivers who actually have to memorize a map of London to get their taxi cab license, they have larger brain regions devoted to spatial or mapping memories. Now, the last way that your brain can change to support learning is by altering its function. As you use a brain region, it becomes more and more excitable and easy to use again. And as your brain has these areas that increase their excitability, the brain shifts how and when they're activated. With learning, we see that whole networks of brain activity are shifting and changing. So neuroplasticity is supported by chemical, by structural, and by functional changes. And these are happening across the whole brain. They can occur in isolation from one another, but most often they take place in concert. Together, they support learning, and they're taking place all the time. So I've just told you really how awesomely neuroplastic your brain is. So why can't you learn anything you choose to with ease? Why do our kids sometimes fail in school? Why, as we age, do we tend to forget things? And why don't people fully recover from brain damage? That is, what is it that limits and facilitates neuroplasticity? And so this is what I study. I study it specifically how it relates to recovery from stroke. So recently, stroke dropped from being the third leading cause of death in the United States to be the fourth leading cause of death. Great news, right? Well, it actually turns out that the numbers of people having a stroke has not declined. We're just better at keeping people alive after a severe stroke. It turns out to be very difficult to help the brain recover from stroke. And frankly, we have failed to develop effective rehabilitation interventions. The net result of this is that stroke is the leading cause of long-term disability in adults in the world. Individuals with stroke are younger and tending to live longer with that disability. And research from my group actually shows that the health-related quality of life of Canadians with stroke has declined. So clearly we need to be better at helping people recover from stroke. And this is an enormous societal problem, and it's one that we're not solving. So what can be done? One thing is absolutely clear. The best driver of neuroplastic change in your brain is your behavior. The problem is that the dose of behavior, the dose of practice that's required to learn new and relearn old motor skills, it's very large. And how to effectively deliver these large doses of practice is a very difficult problem. It's also a very expensive problem. So the approach that my research has taken is to develop therapies that prime or that prepare the brain to learn. And these have included brain stimulation, exercise, and robotics. But through my research, I've realized that a major limitation to the development of therapies that speed recovery from stroke is that patterns of neuroplasticity are highly variable from person to person. Now, as a researcher, variability used to drive me crazy. It makes it very difficult to use statistics to test your data and your ideas. And because of this, medical intervention studies are specifically designed to minimize variability. But in my research, it's becoming really clear that the most important, the most informative data that we collect is showing this variability. So by studying the brain after stroke, we've learned a lot. And I think these lessons are very valuable in other areas. So the first lesson is that the primary driver of change in your brain is your behavior. So there's no neuroplasticity drug you can take. Nothing is more effective than practice at helping you learn. And the bottom line is you have to do the work. And in fact, my research has shown that increased difficulty, increased struggle, if you will, during practice actually leads to both more learning and greater structural change in the brain. The problem here is that neuroplasticity can work both ways. It can be positive. You learn something new and you refined a motor skill. It also can be negative, though. You forgot something you once knew. You become addicted to drugs. Maybe you have chronic pain. So your brain is tremendously plastic, and it's being shaped both structurally and functionally by everything you do, but also by everything that you don't do. The second lesson we've learned about the brain is that there is no one-size-fits-all approach to learning. So there's no recipe for learning. Consider the popular belief that it takes 10,000 hours of practice to learn and to master a new motor skill. Now, I can assure you it is not quite that simple. For some of us, it's going to take a lot more practice, and for others, it may take far less. So the shaping of our plastic brains it's far too unique for there to be any single intervention that's going to work for all of us. And now this realization has forced us to consider something called personalized medicine. So this is the idea that to optimize outcomes, each individual requires their own intervention. And the idea actually comes from cancer treatments. And here it turns out that genetics are very important in matching certain types of chemotherapy with specific forms of cancer. My research is showing that this also applies to recovery from stroke. So there are certain characteristics of brain structure and function that we call biomarkers. And these biomarkers are proving to be very helpful in helping us to match specific therapies with individual patients. And the data from my lab suggests it's a combination of biomarkers that best predicts neuroplastic change and patterns of recovery after stroke. And that's not surprising given how complicated the human brain is. But I also think we can consider this concept much more broadly. Given the unique structure and function of each of our brains, what we've learned about neuroplasticity after stroke applies to everyone. Behaviors that you employ in your everyday life are important. Each of them is changing your brain. And I believe we have to consider not just personalized medicine, but personalized learning. The uniqueness of your brain will affect you, both as a learner and also as a teacher. And now this idea helps us to understand why some children can thrive in traditional education settings and others don't. Why some of us can learn languages easily, and yet others can pick up any sport and excel. So when you leave this room today, your brain will not be the same as when you entered this morning. And I think that's pretty amazing. But each of you is going to have changed your brain differently. Understanding these differences, these individual patterns, this variability and change, is going to enable the next great advance in neuroscience. It's going to allow us to develop new and more effective interventions and allow for matches between learners and teachers and patients and interventions. And this does not just apply to recovery from stroke. It applies to each of us as a parent, as a teacher, as a manager, and also, because you're at TEDx today, a lifelong learner. Study how and what you learn best. Repeat those behaviors that are healthy for your brain and break those behaviors and habits that are not. Practice. Learning is about doing the work that your brain requires. So the best strategies are going to vary between individuals. You know what? They're even going to vary within individuals. So for you, learning music may come very easily, but learning to snowboard, much harder. I hope that you leave today with a new appreciation of how magnificent your brain is. You and your plastic brain are constantly being shaped by the world around you. Understand that everything you do, everything you encounter, and everything you experience is changing your brain. And that can be for better, but it can also be for worse. So when you leave today, go out and build the brain you want. Thank you very much.
All right, so let me ask you guys a question. What, if anything, do you all do to help you pay attention and take in information when you are doing something like watching that video? Does anybody want to share?
I always handwrite my note. I always like to write things down on a piece of paper with a pen or a pencil. It's just I like to do that.
So you write words down, Erin?
Yeah, I'll just like take little notes. Like I just jotted some stuff down. I try sometimes not to, but then I find myself really wanting to because
Yeah.
David and I had it. Yeah, like David's like maybe you shouldn't do that. Maybe you should just listen. I'm like I feel like it helps me listen. So I don't know. And I prefer to write than do it on the computer. And everybody else likes to do the computer, but there's something about the paper and the pencil.
Anybody else?
Um,
Strategy?
Sometimes I'll like doodle a lot.
Yeah.
I know that sounds like I'm not listening, but I'll just like doodle things in the corner of my page if it's not something I need to actively take notes on, and that helps me like, you know, occupy my hands so I can focus my mind.
When you're doing that, Claire, are you drawing something that has something to do with what they're talking about, or just little spirals and kind of random stuff?
It's like a mix. Like if it's like a word that pops out at me or something, or like if there's like a logo on the screen that I see, I'll doodle that. It
That's
just kind of
interesting.
depends.
Yeah.
Yeah.
Yeah. Anybody else want to share? Well, I'll share mine. Mine's kind of a combination of Erin's and Claire's, and I probably have to, let's see, may have to stop sharing my screen. I hate it when it doesn't show me the options. Okay. So I don't know if you can see this because I'm on green screen now. If I put it really close, maybe. Um, so I just kind of did it in the corner of the paper right now. Hold on, I'll see if I can not have my
Yeah, Dr.
background.
Brings we see the beach.
Okay,
Oh,
is
now the
that better? Now it's some green thing. I'm trying not to have a video, a background. Let's see here. I got it.
nope.
Now nothing.
Well, it's like a full background. Okay.
Okay, well, I'll just tell you, it's I drew a picture. I started off when they were talking about juggling, and I drew just a little stick figure that's juggling. And then I kind of did main concepts like long-term memory versus short-term memory. I'll post this after just as a point of interest. It's something I recommend, and there actually is some research out there that talks about, for some people, writing things down, drawing it out, sketching, and sometimes maybe it's something that's not related to what's being said, but sometimes it is kind of sketch note taking that will help keep you on track when you are listening to or watching something. Because I know in this course there's a good bit of that. And like what you had to do for section 2, I know sometimes it gets kind of confusing as far as how do you pick out the key points of that? I'm with what Katie says. If I'm not writing something down, I'll often catch myself thinking about, like, look how dirty my floor is. I should, or something else. Especially when I'm in my home environment, for sure. So doodling is good. Writing things down, a combination of that. Sometimes, and definitely what the research shows is doing something by hand rather than typing helps it get into your long-term memory better for most people. I mean, like the video said, that's, it's different from learner to learner, but it's something that we may want to think about when we're setting ourselves up for success to learn. So I wanted to point that out. And with that, I am going to share my screen again. And let's talk about, let's see here. No, I think I just got off of that. All right, one second. Technical difficulties for me today. Okay, so let's kind of go through the slides that we're looking at now. We're going to start off talking about learning, memory, and emotions. And as our topic for today, pull it up. And I'll tell you that the learning objectives for today I put on the note-taking guide. Really, I want you to, we're going to start off talking about the cranial nerves that we didn't have time to finish last Wednesday, and also the blood-brain barrier. And then we'll really pick up talking about what really has a lot to do with all three of those topics, and that is the limbic system. So that's kind of our target for today. And that is not going to work. I'm going to actually have to show I was trying to show those slides because I think it comes out clearer sometimes, but I'm going to actually show the PowerPoint. So one more time for me to adjust. Okay. Because I do have some videos embedded, which I tried to do so I won't be having to go back and forth today as much. Okay, so let's watch this video.
The cranial nerves consist of 12 pairs of nerves originating directly from the brain and brainstem. They supply both motor and sensory information from the brain to other parts of the body, primarily supplying the region of the head and neck. The nervous system is divided into the central nervous system, so the brain and spinal cord, and the peripheral nervous system, which is further divided into the somatic and the autonomic nervous systems. Broadly speaking, the nervous system is split into an afferent and an efferent division. The afferent division brings sensory information, like vision, smell, touch, and proprioception, from the outside world into the brain. On the other hand, the efferent division brings motor information from the brain to the periphery, ultimately resulting in contraction of skeletal muscles to trigger movement through the somatic nervous system, as well as the contraction of the smooth muscles to trigger activity of the glands and organs through the autonomic nervous system. Now, neurons are the main cells of the nervous system, and they're composed of a cell body, which contains all of the cell's organelles. And when there's a group of neuron cell bodies found in the central nervous system, the whole thing is called a nucleus, while the group of neuron cell bodies that are located outside of the central nervous system is called a ganglion. Neurons also have nerve fibers that extend out from the neuron cell body. And these are either dendrites that receive signals from other neurons, or axons that send signals along to other neurons. Where two neurons come together is called a synapse, and that's where one end of an axon releases neurotransmitters, further relaying the signal to the dendrites or directly to the cell body of the next neuron in the series. Now, the peripheral nervous system consists of nerves, which are enclosed bundles of axons that connect the central nervous system to every other part of the body. Some nerves are purely sensory or purely motor, while most nerves are mixed. Nerves are either classified as cranial nerves, which are ones that exit from the skull and innervate the head and neck, or spinal nerves, which exit from the spinal cord and innervate the rest of the body. And we're going to focus on cranial nerves. There are 12 cranial nerve pairs that are numbered based on the order in which they arise from different nuclei in the brain, except for cranial nerves 11 and 12, which are inverted. In order from 1 to 12, the cranial nerves are called olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory, and hypoglossal. And a quick way to remember them is with the acronym, an old Olympus's towering top A, Finn van German, viewed a hop. The cranial nerves exit the skull through different openings or foramina. The primary functions of the cranial nerves, so sensory, motor, or both, or mixed, can be remembered with the acronym, some say merry money, but my brother says big brains matter more.
All right, so that's just kind of an overview of the cranial nerves, and we're going to talk a little more about that, and I'll give you a couple of other hints of how to remember those. This is important for an OT to know. You will see in medical charts, sometimes people will say this person has paralysis coming from infection of cranial nerve 7, or there's a problem with the cranial nerves that innervate the tongue that make it unsafe for this person to have liquids. And so you need to know what those are. And you can use that mnemonic. You can make up your own. You can straight out memorize it, or whatever you choose to remember those. But know that all cranial nerves do come from the brain, and they exit through openings called the foramen in the skull into the spinal cord, and then go out to innervate other muscles or muscles elsewhere in the body, depending on what their function is. So we'll kind of circle back to a number of these as we learn about different systems. You guys have already, if you did session 2 material, you already learned about some of these that have to do with the sensory systems, and we'll be talking about more of them as we progress through this course. It is, again, something that is important for OTs to know about in clinical practice. And you need to know what type of nerve each of these are. And so that's the thing about the sensory or motor or mixed. And then you need to know just in general what type of muscles, what's the function of the muscles that this nerve innervates there. All right, so here is, let's think this is going to play. Yeah.
GoGuardian Teacher provides a digital window into student online activity, so teachers can see what students are doing on their devices and whether their students are off task or need help. With GoGuardian Teacher, you can push tabs out to students, getting everyone on the same page quickly and cutting down on.
Okay, this is finally the video that I promised. This is the video on the functions of the cranial nerves. Before going forward, keep in mind that this is a very basic review on the functions of the cranial nerves. It is in no way exhaustive of their functions, but this video should give you a basic idea of what these cranial nerves actually do. So let's get started. Some of you have probably seen a picture similar to this to help people remember the functions of the cranial nerves. This is going to be the one that I will be going over, since it is also the picture that I used when I was a student. So let's start drawing this thing. With our blank space, let's start by drawing the number 7. And then again, backwards, let's draw the number 7 again. This, of course, will be the outline of our face. From here, I like to draw in the number 1 as the nose, large 2s as the eyes, and 3s for eyebrows. Next, I like to draw in small 4s at the medial corner of the 2s, and then a very large 5, which will give us part of the mouth and the forehead wrinkle. At this point, I draw in 6 on both sides of the 2s. We could skip 7 here since that is the first thing we did. So we could skip that and draw in 8s as our ears. Now this is where it could get a little tricky, but just follow along and I will explain all of this later. So go ahead and draw in a large tongue and put in another 7 here, but make sure the 7 takes up more than half of the front of the tongue. After that, you could draw in a 9 and then a 10 right behind it. And why don't you give our man or woman a tongue ring? This is where we could draw in cranial nerve number 12. Finally, we're left with cranial nerve 11. You could place them here where our person's shoulders are supposed to be. And that's it. Okay, so let's go over this. Cranial nerve 1, the olfactory nerve, is purely a sensory nerve responsible for the sense of smell. That is why we had placed it as our person's nose. Cranial nerve 2, the optic nerve, is also purely a sensory nerve. that is responsible for the sense of sight or vision. That is why it is our person's eyes. Cranial nerve 3, the oculomotor nerve, like its name says, is a motor nerve for the eyes and aids in eye movement. But keep in mind that there are other cranial nerves that are also responsible for eye movement, which I will talk about in more detail in another video. But just for the sake of simplicity, just remember that cranial nerve 3 aids in the movement of our eyes. Also, I like where we place our cranial nerve 3 because it helps us remember a few of the other things that cranial nerve 3 does. Because it is above our eyes on our drawing, this reminds us that one of the nerve's functions is in opening our eyelids. And because of its proximity to our eyes, you could probably guess that this nerve has other functions associated with our eyes. These are a little more difficult to remember, but include pupillary constriction and lens accommodation, or in other words, it helps with focusing on things we see at different distances. Remember that I said that there were other cranial nerves responsible for eye movement? Well, let's talk about them now. These other nerves are cranial nerves 4, the trochlear nerve, and cranial nerve 6, abducens. Again, for simplicity's sake, just remember that these nerves are responsible for eye movement. And in summary, they are cranial nerves 3, the oculomotor nerve, cranial nerve 4, the trochlear nerve, and cranial nerve 6, abducens. Cranial nerve 5, the trigeminal nerve, is our first sensory and motor nerve. And by looking at our drawing, you could guess that it has something to do with the face. And of course, you're right. Cranial nerve 5 is responsible for facial sensation, or what you feel on your face, like pain or soft touch. You could also include the anterior 2/3 of the tongue in this area that cranial nerve 5 innervates as its sensory component. In regards to the motor component of cranial nerve 5, it innervates your muscles of mastication, and so it helps you open and close your jaw and aids in chewing your food. Cranial nerve 7, the facial nerve, is also a mixed sensory and motor nerve. Its motor component is responsible for facial expression and facial movement, so things like frowning and smiling are all due to cranial nerve 7. Its sensory component, on the other hand, is responsible for taste on the anterior 2/3 of the tongue. This is why we also have it located on the tongue on our drawing. However, don't confuse this with cranial nerve 5's involvement with somatosensation to the anterior 2/3 of the tongue, which is a different type of sense. While we're here talking about the tongue, let's just go ahead and examine it further. Cranial nerve 9, the glossopharyngeal nerve, is both a sensory and motor nerve. Its sensory component is responsible for the sensation at the posterior 1/3 of the tongue. This includes both taste and somatosensation. Note that cranial nerve 10 is also in this area. That's because it's involved in taste as well, but it's actually responsible for taste sensed at the epiglottis. Cranial nerve 10, because it's so far back in the throat on our drawing, also aids in swallowing, but also has functions when we talk and cough. Lastly, on the tongue is cranial nerve 12. I placed it at the tip of our tongue because when a person sticks out their tongue, moves it side to side, you know that cranial nerve 12 is intact because it's responsible for tongue movement. Cranial nerve 8 is literally and figuratively our ears, and it aids in hearing and balance. And finally, cranial nerve 11 we had placed at our person's shoulders because it innervates your trapezius and sternocleidomastoid muscles, which helps with shrugging your shoulders and with turning your head.
I'm not a writer.
Okay.
All right, so let's go. I think that drawing is so helpful, and let's go back to the PowerPoint for that. So if learning the cranial nerves, I would learn the mnemonic and I would practice with that drawing. And to me, that's the best way. You may find that one or both work better for you, but to me, that's the best way to learn that particular bit of information. All right, so let's talk about the blood-brain barrier. And I have this picture there because when I think about the blood-brain barrier, which is is sometimes abbreviated with 3 uppercase B's. It's kind of like a bouncer in a nightclub. It is literally a membrane that is between the spinal cord and the brain that functions as a filter to keep toxins and other things that usually we don't want to get into the brain out. So we've known about the blood-brain barrier for over 100 years. It is permeable, meaning some of the things that can get through it are water, oxygen, carbon dioxide, and even some other soluble substances. We won't get into the details of that, but one thing that makes it tricky sometimes for people with brain cancer to get chemotherapy is this blood-brain barrier. And sometimes they will have to get special medication that helps the chemo get through that blood-brain barrier, or they'll even have to get a catheter inserted into their brain to feed that chemotherapy straight into the brain because of this blood-brain barrier. But in a healthy individual, some of the things that can break down this blood-brain barrier are high blood pressure or hypertension, lead poisoning, sometimes an infection, so like meningitis, inflammation, and trauma to the brain, or to that specific membrane. So when that occurs, it opens up the brain to those toxins and potentially pathogens that can cause inflammation, infection, and a whole lot of other problems. We'll learn more about that specifically in the next neuro course, but I did want to introduce you to this, the structurally and functionally in a healthy human being. So now let's get into today's material, which is the neuroscience of learning, memory, and emotions. And that we're really going to focus on the limbic system. So let's talk about that specifically. Hold on, let me share that again because it was telling me that it was paused. Okay. So the limbic system is located very deep within the middle of the brain. And when you think limbic system, I want you to think emotion, especially very raw emotion, the emotions that haven't been processed or thought about a lot. When we think about emotions, when we're pulling them up from a place of logic or things that we're thinking about, that really is coming more through our frontal lobe. And today we're really talking about what just is your gut feeling, your true emotions. And so the other things that are impacted there and really controlled, at least to some degree, by your limbic system are your memories. That's where your long-term memories, especially those with a strong emotional component, are stored. So on your note-taking guide, I have some specific components or structures that are within that limbic system that I want you to pay attention to. There are more than those, but these are the main ones. And so one I want you to really know is the amygdala. It's kind of a funny word. It looks like it's Amy something, but you pronounce it amygdala. And we're going to talk about that one a good bit. It really has a lot to do with fear and anxiety and memory and attention. So we'll talk about that in just a minute. Some of the other structures that are involved or included in the limbic system are the thalamus, the hypothalamus, the hippocampus, and the corpus callosum. So again, limbic system and emotions, memory, some other things that are also in there is your behavior, and that includes sometimes your character traits, how you treat other people, your ability to empathize, and your motivation. Another thing that's controlled in large part by the limbic system is your sleep and wake cycle. So let's take a look at a drawing that shows some of those structures that I was just talking about. One in there that I didn't mention just a minute ago that you need to know is called the cingulate gyrus. And this one has lots of connections to many other structures within the limbic system. So think of it kind of like Interstate 240, that it connects to itself, it connects to other things right around it, smaller, larger. It plays a big role in decision making when it comes to how somebody is going to respond to sensory stimuli. So the way I can explain that best, I think, is so think about what you learned about in session 2 in regards to sensory stimuli, and let's think about the last part of that, which was pain. If you are doing something, maybe you're going on a walk later today and you stub your toe. There are some choices in how you can respond to that. Now, you know that that feeling of pain, which is really modulated elsewhere in your brain, is connected to your limbic system and your emotion center because of things that have happened in previous experiences in your life. And so your decisions there, which come through your cingulate gyrus, are you going to cry? Are you going to say a bad word? Are you going to ask for help? Are you going to act like it didn't happen? Maybe you're embarrassed. Are you going to laugh it off? How are you going to respond? And that's where that decision comes through. So another big part of the limbic system that you need to know is called the parahippocampal gyrus. What that does is it transmits information from the hippocampus to other areas in your cerebrum, especially the frontal lobes. And so really what it's doing in making those connections is to compare what's going on currently to what has happened in your past. And when it recognizes patterns, it helps you know how to behave. Maybe you're going to behave the same way you did in the past. Maybe something you learned that wasn't the best choice you could make, and you're going to go a different way in your behavior this time. That all is decided through the parahippocampal gyrus. Now let's talk again about the amygdala. The word amygdala actually comes from a Greek word that means almond, because the amygdala is shaped like a pair of almonds. Again, you can see in this drawing, it's very, very deep within the brain. It controls emotions and connects emotions with memory. it is the center of what we learn, even in elementary school, is our fight or flight response system. So it really is necessary for our survival. Again, it plays a primary role in the experiences of anxiety and also understanding social cues and empathy. So if I told you that we have seen in research that there is a difference in the structure, so the size of the amygdala in many individuals with autism, I want you to give me a thumbs up if you think people with autism have bigger than average amygdalas, or a thumbs down if you think people with autism have a smaller than average amygdala. Take a guess. All right. Well, I appreciate you guessing. Yep. You got to go out on a limb for that. So I will tell you that it has been shown to be significantly smaller in people with autism. Not all people, but many or most. So the amygdala, that's one reason that we know that it does help to regulate or facilitate responses to social cues and empathy, because that's something that often is a challenge area for people with autism. Another thing the amygdala does that's important is it serves as a critical component of the brain's reward system. So it is also linked to addiction. So that's what we need to know about the amygdala. All right, moving on, let's talk about the olfactory bulb and tract. Another name for those two things together is cranial nerve number 1. And you should have learned in session 2 about that, but I want to talk about it again today because it's really important, especially in the context of what we're talking about today. Cranial nerve 1, so that olfactory bulb and tract, connects straight to your limbic system, and then it goes to your hippocampus. So and it is really responsible for smells, interpreting smells. And so think about that. When you take in a smell, it goes into your olfactory bulb, up the olfactory tract, your cranial nerve 1 is taking it in, and then sorry, go ahead.
You have some questions in chat.
Okay, yeah, hold on, let me get that. Thank you. Oops. Sometimes it depends how I have my screen arranged, if it lets me see that or not.
Want me to read them to you?
Yeah, if you don't mind, Megan. For some reason, I can't. It's not.
Yeah, no problem. I just didn't want to interrupt. I didn't know if you had seen them. I didn't want to interrupt your lecture. So the first question was, what is an unprocessed emotion? Is this something for little kids learning about emotions or as adults?
Good question. So unprocessed, substitute in the word raw emotion. So that's when, I don't know if you've ever seen somebody experience really very raw grief when they find out, and then this definitely can be an adult as well as a child, somebody who finds out really devastating news. Or if you yourself have heard news that feels like a punch in the gut, or it literally makes your heart hurt because it feels so raw to you. You can't even really explain it or verbalize about it to somebody else. That's what I'm talking about. So it could pertain to kids that don't really know how to put a label on their emotion yet. But it also does pertain to adults. So it's the emotion that you feel before you've really thought about it. You know what? Now the chat box just came up. Okay, so have they studied the size of people's amygdala in people with addiction? And yes, they have. And it actually doesn't appear to be a different size. But what does appear to be different there in some people is the number of synapses or even neurons there, or dendrites. It's more action packed in some people with addiction. In many people, though, addiction is as much of a chemical issue as a structural one, and so it is harder to measure. And if they did say an MRI on all say 10,000 or 100,000 people, they wouldn't probably be able to predict who amongst those people might have problems with addiction, because addiction is so complex. It really, and it's so chemical that and really, as we're talking about the neurotransmitters next week, we'll talk about this more. We're in fact, in session 12, we're going to talk about addiction specifically. So it doesn't appear to be a structural thing, especially in regards to the size of the amygdala. Yes, I can make the notes section bigger too. In fact, I'll just share this whole thing on Blackboard so you guys can see. Okay, so let's talk about, just another minute, the olfactory bulb and tract. So it goes in through your nose, goes up, and it goes into the amygdala. And remember, you think amygdala, emotion. So the first thing that happens right there when it hits the brain is it connects with some kind of emotion. Now, right after that, it goes to the hippocampus, and that's where your long-term memories are. So think about that: smell, emotion, long-term memory. And now what I want you to think about, and I'm going to invite you to share, is what is a smell that you can be exposed to today that brings about very automatically a memory to your brain? Something maybe from your childhood. Does anybody want to share?
I'll share about mine. It's not from my childhood, it's recently, but Chattanooga has very poor sewage system and you can smell it anywhere you walk.
So if you are somewhere and you smell that, it makes you think about being in Chattanooga when that's going on?
Yes.
Yeah, okay. So Shelby says in the chat box, peanut butter cookies and her Nana in a big bowl. And Sam says Old Spice Swagger deodorant and Hawaii. All right, yes. Sometimes we can have alcohol relate that to, like Fireball. I know someone who, and it's not me. I'm not saying like I have a friend, but I know someone who can't stand the smell of limes because of a gin and tonic incident from 30 years ago. Yeah, I think Fireball is it for a lot of people. Buttercups reminds Alex of her grandmother's house. Cigars and cigarettes sometimes will bring that. Sometimes we just think, oh, that doesn't smell good. But sometimes a smell like that can be something that smells good to us because of this exact system. Shaving cream powder, Allie says, oh, and even though her dad used it and she thought it was horrible, maybe it's kind of a good thing to smell every once in a while because it reminds you of that fond memory. Sunscreen and the beach. perfume and certain people, mom or grandmother or cologne, and somebody, Kayla says asphalt and Silver Dollar City. That's kind of funny. The perfume love smell reminds Amanda of middle school. That's also kind of funny. I've had people say baking bread and, you know, that kind of thing before, for sure, that they remember things like that. I'll tell you one of mine. Probably about a dozen years ago, I fostered a 4 newborn skunks. And just before you freak out, just so you know, skunks don't develop their scent glands so that they can spray until they're 6 months old. So they were really cute, and they had a little bit of a smell, but it was faint, and they couldn't spray. And we kept them for a couple of months, and until they were ready to release. Their mom got run over, and they couldn't eat on their own, or, you know, that any of that kind of thing. So, but to this day, even this many years later, whenever I smell a skunk that has sprayed, it reminds me of those sweet little baby skunks. So I kind of like that smell, which I know is really kind of weird. Shelby says Hollister perfume. Oh, Barks root beer reminds Allie of her grandmother because her grandmother always had it at her house. Isn't it funny the way a number of us say our grandmother? And why is it that smells connect to, maybe more than any other family member, to our grandmothers? That's kind of interesting. Joe says freshly cut grass, Friday work days with her dad. I have one freshly cut grass, reminds me of running cross country in high school. So I always like to smell that. Yeah. All right. So we have a lot there. You are welcome to keep putting those in the chat box if you want. But if you've ever wondered why that is, whether that memory is good, like, you know, peanut butter cookies, or for me, skunks, or if it's bad, like Fireball, then it's because of your limbic system and cranial nerve 1 that takes in that smell. It hits your amygdala and then goes to your hippocampus. And so your amygdala with those emotions and your hippocampus with your long-term memories, those are directly linked. So that's something important to know. So one more thing, and then we're going to play a Kahoot, and then we will take a few minute break. The hippocampus. Now, it is located in the parahippocampal gyrus. So that's just the section. If you look at that slide, the hippocampus is that purple area in the slide. It's not really purple. And interestingly, if you look at the coronal section, so if you slice things in the way that it is here, it looks a little bit like a seahorse. And that's what hippocampus means in Latin. So that's where it got its name. And again, like I was just saying, when you think hippocampus, one thing I want you to think is long-term memory, especially long-term memories that have some kind of emotional tie to it. So if you were really happy, if you were really traumatized, if you were really sad or excited or surprised, those memories are definitely going to be stored in your hippocampus and will be easier to pull up than memories that weren't as emotionally connected. So also important, and I think interesting to note, that throughout life, for sure, neurogenesis, so that means the birth or development of new neurons and neuronal connections, occurs in the hippocampus. So what that tells us, and really what that explains, is why sometimes, or actually very often, when the brain is injured, so maybe it's through a traumatic brain injury like a car accident or a gunshot wound to the head or a stroke, that the long-term memory is often still there. We see this in dementia also. If you've been around somebody with Alzheimer's or any other kind of dementia, you might have noticed that they can talk about their childhood or things that happened a long, long time ago that they remember. But sometimes, I mean, very often they don't know what day it is. They don't know what they ate for their last, their most recent meal. Like, what did you eat an hour ago? And that is because of the hippocampus. Whereas your shorter term memories are more in your frontal lobe. And so that degeneration of that starts happening sooner in the front of your brain with dementia. And you have a stronger, more solid link and more neurons that continue to form in that hippocampus when there is an emotionally connected long-term memory. All right, so let's stop with that and let's play a Kahoot to kind of jazz things up again, and then we'll take a few minute break. Let me get to that. I do have the Kahoot link, and it will release for you guys when this class session is over, so you should be able to get straight to it when you are studying or reviewing or doing some fun retrieval practice. All right, so let me share that so you can get the number. $407-5195. Oops. I like to put it on reggae.
Thanks everyone for the HPD shout outs and your names. Thanks.
That's fun. All right, here we go. 10 questions. Some retrieval practice. What is the basic building block of the nervous system? It is the neuron. About how many neurons are in the human nervous system? $90 billion, or you might see 100 billion at some places, but if you're given the choice that are here, choose 90 billion. That's a lot. What's the name of the protective covering around neuron fibers? Good, most people got that. That is the myelin. So remember, it's sometimes called the myelin sheath. What term refers to the brain's ability to reorganize itself by forming new neural connections? It is neuroplasticity. Everybody got it? Or the majority? Here we go. Other than the central nervous system, what's the other main division of the human nervous system? It's the peripheral. So we've got the central and the peripheral nervous system. If you remember that graph that was on your handout last time, those are the two big headings. What is the small pitted area in the center of the retina of the eye? The fovea. Very good, from session 2. What part of the eye turns light into electrical signals? Photoreceptors. What are two parts of the body with the highest concentration of touch receptors? Fingers and lips. What type of sensory fibers respond to stimuli that indicate pain? Nociceptors. What part of the brain is primarily responsible for processing visual information? The occipital lobe. Here's our podium. All right, good job, everybody. All right, so let's take 5 or 6 minutes. Let's come back at 2:15 and we'll bring it on home.
So we want to have a serious conversation.
Yes.
Because we checked.
Structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons. all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.
So thanks to HM, in part at least, scientists now know that the medial temporal lobe, so remember the temporal lobe is kind of above your ears and a little bit to the back of your head, and that's where the hippocampus and the parahippocampal region is, that we know that that works with other regions of the brain to organize and consolidate memories. There's several different kinds of memories, and they're processed by different areas of the brain. And we're going to talk about some of those in this course, and you'll learn about some of those and problems that go along with some of those in the next neuro course. But first, I want you to watch this video, and I want you to take the takeaway I want you to get from this is, what is a motor neuron?
Oh my God, don't let me go. Why do people cry at movies? Why do sports fans get so excited during a game? Why do they feel anxious if their team is losing? The answer? Mirror neurons, a set of brain cells on either side of the head that might explain why people feel empathy.
What we found is the mechanism that underlies something which is absolutely fundamental to the way that we see other people in the world.
The mirror neuron was first discovered at the University of Parma in Italy. Researchers there identified a neuron that fires both when a monkey picks up a peanut and when the monkey watches someone pick up a peanut.
The same neurons, one neuron, fire both when the monkey observes something and when monkey is doing something. It's almost unbelievable.
It was surprising because this cell which was involved with motor planning for the monkey turned out to be interested in the movements of other people as well.
The brain uses the mirror neurons to mirror the movements it sees. And this happens in people too.
If you can use the years of training that you yourself have done, learning to crawl, then learning to walk, then learning to eat, this is an incredibly rich set of knowledge that you could apply to the problem of actually seeing what's going on. The mirror system is the way that you tap into, the way that you harness your own abilities and project them out into the world.
So sports fans can picture themselves playing when they watch a game. But what makes them feel the pain when their favorite player drops the ball?
So the idea was to try to figure out how the emotional system and this motor system are connected together.
To find out, Marco Iacoboni looked at the brain scans of individuals when he presented them with a series of faces. In the first round, he asked people to imitate the faces. In the second round, he asked people to simply look at the faces. As expected, he found that the mirror neurons were activated during both rounds. But in addition, he found that the happy area of the brain lit up whenever people either imitated or looked at happy faces. So he thinks the mirror neurons can send messages to the emotional system in our brain. And that's the mechanism that allows us to feel empathy with other people.
We strongly believe that's a unifying mechanism that allows people to actually connect at a very simple level.
So what happens if the mechanism is broken?
It's been known for some time that children with autism could be quite intelligent, but have a profound deficit in social interaction.
Christian likes to play, but like many kids with autism, he prefers to play alone. He has a hard time communicating and does not always understand questions.
So Christian, can you tell me what you did in school today?
Doing well.
Dr. Ramachandran and his graduate student hope to gain understanding of autism by studying the brainwaves of children like Christian.
So we're going to be reading your brainwaves with this cap.
When mirror neurons are working correctly, they fire both for doing an action and seeing the same action. So for most people, the brainwaves for doing and seeing look the same. But for Christian, the waves look different.
Their brains may indeed be different in that regard, and they may have deficits in the mirror neuron system. But we don't know this for sure yet, and additional work needs to be done using brain imaging.
What we do know is that healthy humans are very social.
Everybody's interested in this question. What makes humans unique? You can say humor, you're the laughing biped. Language, certainly, okay? But another thing is culture. And a lot of culture comes from imitation, watching your teachers do something.
So it sounds like the mirror neuron is a big part of what makes us human.
There would be very little point in having a mirror system if you lived on your own. There'd be a lot of point in having a digestive system if you lived on your own. There'd be a good point in having a movement system if you lived on your own. There'd be a good point in having a visual system if you lived on your own. But there'd be no point in having a mirror system. The mirror system is the most basic social brain system. It's a brain system which there's no point in having if you don't want to interact or relate to other people.
All right, so hopefully you picked up from that what a mirror neuron, I can't talk, is. It's really something that fires when an animal or a human acts and when you observe somebody else doing that same thing. And so it
your children moving, so I was like, okay, oh, you're back.
Sorry about that. I guess my internet dropped off. That was so inconvenient. So anyway, sorry about that. All right, so go back to it. Okay, so a mirror neuron is one that lets your brain experience something as if you are doing it, even if you're just observing it, really. So that's why it's called mirror. All right, so let's talk about the different types of memory. The first one, and the one that in the book it really talks about the most by far, is declarative memory. And this one is called declarative memory. Let me make that chat section bigger, because it's information is stored in those memories that you can declare or describe or tell somebody about. So it's stories that you could tell from things that have happened or things that you've learned or things that you've experienced. So there's semantic memories, and that is things that you have picked up or knowledge that you've picked up, like what words mean and how to do certain math problems, things like that. And then there are episodic memories. Those are both, and that's things that you remember from what you've experienced. And those are both types of declarative memories. And that goes into, of course, your long-term memory, and that is regulated in your hippocampus. Now, in between your short-term memory and your frontal lobe and your long-term memory and your hippocampus is something called your working memory. And that's kind of your holding tank, I think of it as. It still is considered a part or type of declarative memory, but it's not exactly a long-term memory. And so a prime example of that is a phone number that somebody tells you and you're trying to remember it. Maybe you don't have anything to write it down on, and so you're trying to remember it until you can dial it. And maybe you don't even care about remembering it long term, but you have to remember it for a short period of time at least. Remembering directions is kind of like that also. And so that's called working memory. You have to remember it just while you're working with that set of information. That really goes through your prefrontal cortex. Another kind of declarative memory is the spatial memory. And that has really a tie to what you have seen, or especially in regards to where you saw it or experienced it. And so it does have to do with the hippocampus, again, with that long-term memory. So those are all part of declarative memory. All right, another short video, and this one is going to talk about the types of memory.
Think back to a really vivid memory. Got it? Okay, now try to remember what you had for lunch 3 weeks ago. That second memory probably isn't as strong. But why not? Why do we remember some things and not others? And why do memories eventually fade? Let's look at how memories form in the first place. When you experience something, like dialing a phone number, the experience is converted into a pulse of electrical energy that zips along a network of neurons. Information first lands in short-term memory, where it's available from anywhere from a few seconds to a couple of minutes. It's then transferred to long-term memory through areas such as the hippocampus, and finally to several storage regions across the brain. Neurons throughout the brain communicate at dedicated sites called synapses using specialized neurotransmitters. If two neurons communicate repeatedly, a remarkable thing happens. The efficiency of communication between them increases. This process, called long-term potentiation, is considered to be a mechanism by which memories are stored long-term. But how do some memories get lost? Age is one factor. As we get older, synapses begin to falter and weaken, affecting how easily we can retrieve memories. Scientists have several theories about what's behind this deterioration. From actual brain shrinkage, the hippocampus loses 5% of its neurons every decade, for a total loss of 20% by the time you're 80 years old, to the drop in the production of neurotransmitters like acetylcholine, which is vital to learning and memory. These changes seem to affect how people retrieve stored information. Age also affects our memory-making abilities. Memories are encoded most strongly when we're paying attention, when we're deeply engaged, and when information is meaningful to us. Mental and physical health problems, which tend to increase as we age, interfere with our ability to pay attention and thus act as memory thieves. Another leading cause of memory problems is chronic stress. When we're constantly overloaded with work and personal responsibilities, our bodies are on hyper alert. This response has evolved from the physiological mechanism designed to make sure we can survive any crisis. Stress chemicals help mobilize energy and increase alertness. However, with chronic stress, our bodies become flooded with these chemicals, resulting in a loss of brain cells and an inability to form new ones, which affects our ability to retain new information. Depression is another culprit. People who are depressed are 40% more likely to develop memory problems. Low levels of serotonin, a neurotransmitter connected to arousal, may make depressed individuals less attentive to new information. Dwelling on sad events in the past, another symptom of depression, makes it difficult to pay attention to the present, affecting the ability to store short-term memories. Isolation, which is tied to depression, is another memory thief. A study by the Harvard School of Public Health found that older people with high levels of social integration had a slower rate of memory decline over a 6-year period. The exact reason remains unclear, but experts suspect that social interaction gives our brain a mental workout. Just like muscle strength, we have to use our brain or risk losing it. But don't despair. There are several steps you can take to aid your brain in preserving your memories. Make sure you keep physically active. Increased blood flow to the brain is helpful. And eat well. Your brain needs all the right nutrients to keep functioning correctly. And finally, give your brain a workout. Exposing your brain to challenges like learning a new language is one of the best defenses for keeping your memories intact.
Oops. All right, so non-declarative memory, the other kind that we're going to talk about, is also known as implicit or procedural memory. So that's on your handout. And you really just pull that out without a lot of extra effort. So think about that when you do something that has to do with motor skills or even talking, so language that you've learned. You don't have to declare that you know something or that you remember something. You're just doing it. but it's something that you're doing from something you have learned or experienced in the past. HM in that story, he did not lose that type of memory. In fact, he could acquire new motor skills and then remember them later, even though he couldn't really acquire and remember new information. So that tells us, and this is part of the reason why the story of HM is so important, that tells us that different types of memories are stored in different types of the brain. Motor skill learning, like riding a bike or playing the piano or any instrument, or even really walking, it involves a number of areas of the brain, but there are three that are primarily involved, and that is the basal ganglia, the prefrontal cortex, and the cerebellum. Remember, we talked about the cerebellum last week. These three areas are going to be the focus of what we learn about in the next session, in this course, session 4. So you'll hear about this again. All right, one more piece to what we need to learn about today, and that is emotion. So we've already said that emotion is very closely linked and really kind of controlled through the amygdala. Other structures in the brain are involved as well, but the amygdala is a big part of that. Two other areas that are located very near the amygdala, but a little bit anterior to that have also to do with emotions are the insular cortex and the periaqueductal gray. So those three parts of the brain are what we consider to be primarily concerned with emotions. The amygdala helps us deal with motivation and emotional behavior, and it helps us interpret emotions. And like we've said, social cues. Now, something I think is really interesting is the insular cortex, or sometimes it's just called the insula, is where we get the emotion of disgust. So when we were talking about memories and smell, and we were talking about sometimes there are those odors that just really make us cringe that are tied to some long-term memories or experiences of long ago. We know that it's not just the amygdala that's involved there, it's also the insular cortex. And that really is a survival function that our brain does us the favor of having, because it ties together something from our past experiences to something we experienced a negative effect from before, and then our future behavior. So if you drank too much Fireball before and it gave you a vicious hangover, made you feel sick, you're going to remember that. Your insula is going to connect the long-term memory of that and the emotions and physical feelings that you felt, and going to tell you that your behavior should change next time. And that's the way that emotions, memories, and behaviors are tied together. That's one example of that. All right. One more thing that has to do with what we're talking about, and one more video, and I think you're going to really like this one. This is something I want you to think about in the coming days a little bit more. And it's something called daydreaming. You've heard that term before. Sometimes we think of it, it's very akin to something today that's sort of a buzz term of mindfulness. They're not the same, but they're related. And those things have to do with the same areas of the brain really that we're talking about, and also mirror neurons, your memory, and your prefrontal cortex. So watch this video and then we will wrap things up. You're going to see somebody familiar here.
On a November afternoon in 2007, I went for a horseback ride. I rode up a hillside and suddenly the horse began to buck. I flew through the air and landed on my heel, forcing the head of my femur through my hip socket, shattering my hip joint. As you can see, the surgeon put my hip back together, but I was told that I would likely walk with a limp. At the time of the accident, I had been working on my PhD. And during my recovery, I took a break from that and I spent a lot of time daydreaming. In one of those daydreams, I would visualize myself walking across the stage to earn my diploma without a limp, which they had said I would likely have, and wearing heels. In all honesty, I've always been a daydreamer.